Welcome aboard the BioTrain. In this video, we're going to talk about biotechnology and genetic engineering. Biotechnology is the exploration of biological processes by humans to improve lives and make useful products. Humans have been using biotechnology for thousands of years. It was first used in selective breeding and the use of microbes to produce wines, cheeses, and other food products. Selective breeding is a form of biotechnology because humans were manipulating the traits of organisms to suit their needs and improve their lives. Thousands of years before humans understood the mechanisms for reproduction, they understood that if they chose two individuals with desirable traits, those traits tended to appear in the offspring. So by carefully choosing the individuals that they allowed to breed together, they were able to manipulate the characteristics of organisms dramatically. We see this dramatically in the manipulation of dog breeds. All dog breeds are members of the same species, but they have been dramatically manipulated by humans through the process of selective breeding. The Great Dane and the teacup Yorkie are both members of the same species, but they seem like very different organisms. They are so different because they have been manipulated by humans through the process of selective breeding. When the process of selective breeding is practiced carefully and consistently over thousands of generations, you can get dramatic results. All of the domesticated animal and plant species that we take for granted were carefully selectively bred over thousands of generations by early humans. Just like the dramatic results of the different dog breeds that were obtained through selective breeding, dramatic results can be also obtained with plant species. All of the different food crops shown in this slide were derived from one wild mustard species through the process of selective breeding. The importance of selective breeding and how it has improved our lives can't be overemphasized. From cattle breeding in ancient Africa to modern agriculture, selective breeding continues to be an important way that we manipulate living organisms to our benefit. Humans have also understood for thousands of years that they could harness the metabolic activity of certain microbes like unicellular yeast and bacteria to make wine, cheeses, and bread. As human understanding of biology and the world around them increased, they discovered new ways of manipulating organisms for their own good. Specifically, the discovery of microscopic cells and the understanding of biological molecules led to what we will call new biotechnology. This new biotechnology focuses on our modern understanding of DNA, RNA, and proteins. Our modern understanding of DNA and how it leads to the production of specific proteins, which ultimately controls the characteristics of the organism, has led us to this new form of manipulating organisms that we call genetic engineering. It's easy to understand how a carpenter is able to manipulate wood using carpentry tools. But how is it possible to manipulate a DNA molecule which is way too small to manipulate directly using physical tools? The way that humans manipulate DNA is by using protein molecules that naturally interact with DNA in living cells. The main type of protein that we use are enzymes. These enzymes have been isolated from bacteria and are now used as the main toolkit for manipulating DNA. The three main types of enzymes used in genetic engineering are DNA polymerase, which allows DNA to be replicated in a test tube, DNA ligase allows pieces of DNA to be attached together, and restriction enzymes cut DNA into pieces. The ability to cut DNA into fragments, to attach pieces of DNA back together, and to replicate DNA allow humans to directly manipulate the genetic material that leads to the characteristics of all living organisms. So let's take a closer look at some of the tools that genetic engineers use to manipulate DNA. DNA molecules are large macromolecules. For example, the human genome contains 3 billion base pairs. So if you're going to work on DNA, you need the ability to cut and isolate small pieces of DNA. This is where restriction enzymes become an important tool. Restriction enzymes are naturally occurring in bacteria species and are part of the immune system of the bacteria. They cut up foreign viral DNA and make it harmless to the bacteria. When scientists discovered restriction enzymes, they very quickly realized that these enzymes could be used to manipulate DNA in a test tube outside of a living cell. Restriction enzymes do not cut DNA randomly. They cut DNA at specific recognition sites. Each restriction enzyme, and there are thousands that have been isolated from bacteria, have a specific recognition sequence. The enzyme finds this recognition sequence and only cuts the DNA at these sites. For example, the restriction enzyme EchoR1 only cuts at the recognition sequence GAATTC. The red line shows that this enzyme specifically cuts between the G and the A in the recognition sequence on both strands. Here is a diagram showing EchoR1 cutting a piece of DNA at 2 recognition sequences. Notice the enzyme cuts the DNA between the G and the A. EchoR1 makes what's called staggered cuts, which leaves exposed bases called sticky ends. Not all enzymes make staggered cuts. Some enzymes cut straight across from each other. For example, in this diagram, EchoR1, BAMH1, HIND3, and HHA1 all have staggered cuts and make sticky ends, while SMA1 and ALU1 cut straight across from each other and make what's called blunt ends. Restriction enzymes make it possible to isolate specific pieces of DNA, specifically if a researcher is trying to isolate a specific gene. Once the researcher knows the DNA sequence, they can choose the restriction enzymes with the right recognition sequence to cut out that specific piece of DNA. It would not be possible to manipulate DNA without restriction enzymes, just like it wouldn't be possible for a carpenter to manipulate wood without some way of cutting it into the pieces that they need. Cutting DNA into different fragments in a test tube using restriction enzymes would be useless without a technique for separating those fragments by size. This is where gel electrophoresis comes in. Gel electrophoresis is a technique that moves different sized DNA fragments through a gel matrix at different speeds. The best way to understand gel electrophoresis is to think about a running race. In a running race, all the runners start at the same point, and as they move through the running course at different speeds, they separate out. Instead of runners separating over a running course, in gel electrophoresis, different sized DNA fragments are separated by moving at different speeds through a gel matrix. Let's take a close look at this diagram. Beginning on the right, the three small test tubes have DNA plus restriction enzymes. This means that the DNA in those test tubes is being cut into different size fragments by the restriction enzymes. The mixture of different sized DNA fragments is then added into the little wells on one side of the gel. The gel is then placed in a buffer solution that conducts electricity, and a current is run through the gel. Because the DNA fragments are negatively charged, they get pulled through the gel matrix towards the positive end. The shorter DNA fragments move more quickly through the gel matrix and the longer DNA fragments more slowly. So the shorter fragments end up further away from the starting point, and the longer fragments are spaced out before that. Just like in a running race, the fastest runner ends up furthest away from the start, and all the other slower runners are spaced out behind them. I've introduced some of the basic tools that genetic engineers use to manipulate and study DNA. When you put new tools into the hands of humans, they tend to use them in new and creative ways. This is true with genetic engineering. It's a fast-paced field where huge advances are made very rapidly. Because it's a new field, there's a lot of interest and new and creative ways to apply and use these tools. To recap, humans have been using their understanding of biological processes to manipulate and use organisms for their benefit for thousands of years. First, it was through selective breeding and the use of microbes to help make food products. Then, as our understanding of biological processes increased, we began to be able to manipulate the hereditary molecules directly. These new technologies to manipulate life have raised some ethical concerns. We will talk about those ethical issues with biotechnology in another video. And as always, I hope that helps you stay comfortably seated on the biotrain.